Okay, thank you
Thank
very
you.
much. Thank you all for spending your lunchtime with me hearing about the science that my lab is doing in collaboration with Almaden Genomics. Okay, so first we're going to go through a little bit of introduction about big data and the challenges that it faces. So I don't think I really need to emphasize too much in this audience the enormous power of the acquisition of large data sets, whether it be RNA sequencing or mass spec or almost everything else, and how much of an impact that that's having on how people like me, experimentalists, go about approaching our day-to-day projects. You know, from my perspective, these techniques are providing amounts of data that we could not have imagined 10 or 20 years ago. But they're landing in the hands of experimenters who are much more comfortable designing benchtop experiments and who have little or no ability to write code or deal with complex programs or even analyze these large data sets. So we have a sort of paradox here. The scientific opportunities provided by these powerful techniques are enormous, but there's also enormous dangers for wasted effort, misleading interpretations, and irreproducibility. So what's a way of thinking about that? That wet lab researchers right now go and we do experiments. We submit the results for analysis, usually to some kind of core facility. Then it all falls into this black box. And then we get some kind of spreadsheets or something back. And then we do something to somehow analyze this data and get our results. And I think this is the problem right here, is that we really don't understand what's going on. Okay, so specifically, what are these challenges for a person like me? Well, one thing is that the data sets are very large, they're complex, and then overlay that the biological data can be messy. That becomes very difficult to work with. There's also a problem with documentation. We don't have any clear step-by-step guides that can aid in understanding what we're doing or repeating previous studies. There's method uncertainty. Steps can be vague or missing that make it hard to repeat analysis. So that applies to the data that we get in my lab. We don't exactly know how it was generated or what went into it. And it's even worse when we look at published data. We don't really have any clue about what went on there. Okay, finally, you could have different software versions that can lead to different results, and that complicates studying repetition. Okay, so how do biologists address these challenges? Well, I think there's a certain subset of biologists that just ignore them, or maybe I should say are completely ignorant of them. They just go on, they take what they have at face value, and they cherry pick their data, they publish their paper, and that's success for them. It's also possible to collaborate with a computational biologist. And in theory, this is ideal. The problem with this is that the computational biologist, who's highly talented and whose skills are sought after, could make your project their 10th priority, not put a lot of effort into thinking about it, and that leads to time, and it means that your data doesn't get as much of attention as you would want. Finally, you can always analyze the data yourself, but that means growing the expertise in your own lab. And that's a pretty tall order to expect an experimental biologist to end up training as a computational biologist. I think that's probably ideal, someone who's expert at both of those, but I don't think there's too many of those people walking around. Okay, so what are these practical challenges? Well, specifically, the biologists lack experience with coding and statistics. Managing data is messy. We always have a big signal to noise problem, for example. Staying up to date with the latest bioinformatics tools can be challenging or perhaps even impossible if you're an experimentalist. And then there's these collaboration hurdles. So if we want to talk to a computational biologist, that person will know the computation really well. They will have skills that I cannot even imagine, but they may not actually understand what the biological processes are, or even have the vocabulary to talk to us about the biological processes without difficulty. And then finally, we can be limited by the availability of high performance computing. Okay, so what are the consequences? So, to give you my background, I've been a professor at the University of Texas Southwestern Medical Center for 30 years. I'm president of the Oligonucleotide Therapeutics Society, a society that contributed most of the science behind the COVID vaccine and that's producing several approved drugs every year. I'm also an editor for Nucleic Acid Research. I handle about 300 manuscripts a year, and I've been doing that for about the last 11 or 12 years. And one of the things that we see is a lot of misuse of large data sets, a lot of cherry picking, which with the amount of manuscripts that I've been able to see over the years, I can pretty much editorially reject out of hand. But which actually, the more cherry picking there is, the more the tendency is that they actually go to high profile, journals. And it's created a great deal of distrust within the experimental biology community of the kind of things that you guys do, because of the misuse by experimentalists of the tools of computational biology. So that's a real problem that I believe needs to be fixed. So kind of falls into this. One of the consequences is don't try this at home, that these tools are put in the hands of people who either don't understand them or willfully cherry pick the data from them to suit their hypotheses. And that leads to misuse of these powerful computational approaches. The other one is just trust me, that they will produce data. They will produce nice pie charts or heat maps or volcano plots, but they won't really explain how they got there. And if you're not explaining how you got there, and if you're not doing some experimental validation, it really is just trust me, I'm telling you the truth. But I think by about in 2023, I think we've all learned that that isn't enough. And finally, that all leads to a lack of reproducibility. Reproducing a published experiment is a thankless task, and it becomes impossible when protocols are garbled and computational pipelines are not available or poorly defined. I mean, simple test. You're doing a student journal club. If that journal club with students who are extremely bright, if they cannot delve into the data and critically assess it, then there's probably something wrong with the way the data is being explained and presented. Okay, so now I'm going to back up a little bit and tell you specifically what I do, and then how we've been working with Almid and genomics to solve some of the problems that we face. So this is going to be about RNAi, which I think, even after over 20 years since the discovery of RNAi in mammals, is still an unmet opportunity. Okay, so there are two general ways in which we approach gene therapy, gene silencing, if we want to silence a therapeutic gene to make a drug. The first is to use antisense oligonucleotides to bind a messenger RNA that can change splicing or inhibit gene expression. The second approach would be to take a double-stranded RNA. It binds to Argonaute protein, a guide strand then forms an Argonaute guide strand complex, and that, in theory, can recognize just about any RNA sequence in the genome. So you can tell between these two approaches, we have the ability to pretty much bind to any RNA and then possibly affect the course of almost any disease. So tremendous potential there. And it's certainly, as I said, coming to the point where success in the clinic is routine. It's just a question of how efficiently we can go through the drug discovery process and how we can imagine what the disease targets are going to be. Okay, so that's led to a large number, let's say, 15 and counting, approved oligonucleotide and double-stranded RNA drugs. And this is a remarkable achievement from 5 years ago when I believe there was only 1 or 2. Okay, but now let's flip it. Okay, so those are synthetic RNAs or antisense oligonucleotides that target messenger RNA. What about natural microRNAs? So microRNAs are synthetic or naturally expressed synthetic RNAs that bind to RNA and regulate gene expression. So the idea is that this is a natural gene silencing mechanism. Argonaute protects the RNA and facilitates recognition, potential to control almost any gene. And therefore, if we could learn how to bind synthetic molecules to this natural microRNA, we'd be able to interfere with this process. And again, have powerful tools to control gene expression. So this idea of using synthetic oligonucleotides to bind to microRNAs to control gene expression, that's nothing new. People have been looking at that since the 2000s, and a few $100 million has been invested in companies that tried to make drugs that would target microRNAs. Well, how have those fared? Well, that's the number of approved therapeutics that target microRNAs. There haven't been any yet. So, we have to ask ourselves, these microRNAs are supposed to be incredibly powerful. Why have we not been able to get therapeutics for these when we've been so successful with synthetic double-stranded RNAs to target gene silencing? Okay, so clinical success demonstrates the power of RNAi in vivo. All you have to do is go across the river to Kendall Square, visit Alnylam, and you'll see how powerful this approach is. We know that microRNAs have demonstrated roles in plants and animals. That's undoubted. And what's more, if you were just to look at PubMed, you would find that there are thousands of papers, thousands of them, with the keyword microRNA in them every year. So you would think that this is a well-known field where everything is settled and we should be able to go straight to clinical targets and have success. Okay, and this just makes that point. This is the number of papers that have been published that cite microRNAs per year. For example, 6,700 cite cancer and microRNA. So if you were just to look at this number of papers, you would think that this field is well settled and there's plenty of information for a productive drug discovery program, the kind of thing that is going to be very profitable for a lot of companies. All right, unfortunately, first impressions deceive in this case. When you look at these papers closely, what you find is that most of them lack proper controls. In fact, I would say almost all of them lack insight into mechanism. And what's more, there's an insufficient appreciation that microRNA function is complex. Now, as someone who appreciates complexity, I would say it is beautifully complex. This is something that can fill an entire career for the next 30 years figuring out how this works. But if you do not appreciate that complexity, appreciate that it is there, a drug discovery program is not going to advance. The complexity must be treated with respect. Okay, so let me just give you a little bit of a taste of this complexity. A lot of investigators in the microRNA field proceed from the assumption that if you can calculate a small match of a microRNA to a gene, that microRNA is controlling the gene, something like that. However, that almost certainly is not what's happening. Probably what's happening is that you have multiple microRNAs binding next to one another on the gene, forming cooperative interactions that produce an effect that is strong enough to actually control the gene expression. But what's worse is that these are not the same microRNAs. These are probably different microRNAs binding beside each other. So you have to separately make accurate calculations about both. And then what happens is there's another layer of proteins on top of all of that. In this case, the protein TNRC6, which binds to Argonaute protein and actually promotes cooperative interaction. So not only do you have multiple microRNA species that are different microRNAs binding near one another on an RNA, but then you have an extra level of cooperativity on top of that, making the system even more complex to model. Okay, and then underneath that, we have the problem that this is like typical data showing which potential microRNA Argonaute complexes bind at a gene. And what you can see is even when you restrict the microRNAs to only the ones that are the most highly expressed inside a cell. Oh, and don't even get me started with the fact that almost no studies actually appreciate that more highly expressed microRNAs are much more likely to be active than the bulk of microRNAs which are expressed at extremely low levels inside a cell. That it's hard to predict which of these microRNAs is actually going to be the one that's going to have a function. So I think you see how there's complexity here, and that accurate analysis is going to be incredibly important for figuring out what are going to be the experiments that actually have to be done in the laboratory with the limited amount of time we have available. Okay, so this is what we need to figure out. We need to figure out which microRNAs modulate a critical function. We need to figure out which genes are modulated and how does controlling them lead to therapy. And finally, that's all going to lead us to when does a microRNA play a critical role in health and disease? If we can answer all of these questions, then we have a chance, realistically, of having a drug discovery program that might be able to succeed at targeting a microRNA. Okay, so now I'm going to tell you a specific story in my lab about one of my special interests is thinking about how RNAi in the nucleus of cells might be able to affect splicing. All right, so for the last, almost about 16, 17 years now, my lab has been interested in the topic about whether RNAi, which is dogmatically thought to work in the cytoplasm, might also work in the nucleus. Just as an aside, this idea met with a tremendous amount of pushback for many years, but now it's becoming widely accepted that it does happen. And if you think about RNAi in the nucleus, you can now start thinking about, could it affect transcription? Could it affect enhancer elements? Could it affect splicing? A whole new world of gene targets. So we published a series of papers over the last 3 years looking at this issue. And we relied on a variety of cell lines that were knocked out for various RNAi factors so that we could do extremely well-controlled studies. And we found that in normally growing mammalian cells, RNAi does not do that much. And that it's very hard to find really good targets for focused experiments. So what we hypothesized was that maybe these cells need to be pushed by environmental stimuli to go into a state where RNAi becomes more important. So the idea here is that if you stress the cells, maybe that's where this extra level of RNA regulation on top of normal transcription factors and everything else the cells use becomes an adaptive response that the cells need. So we asked the question, very simple question, of we use HCT116 cells. Those are the cell line that we've made all of our knockout cells. What happens when they're grown to overconfluence? Very simple type of cell stress. Okay, so this project is done by a brilliant student in the lab, Crystal Johnson. She looked at cells in normal 2D cell culture, in 3D organoid culture, and in primary tumors. And so what HCT116 cells will do is they will not only grow until they completely fill a cell culture dish, they will start growing on top of one another until they stack about 4 deep. So I'm going to be telling you about the results of cells that are different days of confluence. So they might be at 80% confluence on day 3, 200% on day 5, and maybe 400% on day 7. That's our simple experimental model. So Crystal did this, and what did she find? She found that we saw a shift of Argonaute from mainly being a cytoplasmic protein with some in the nucleus to almost being entirely nuclear in just regular 2D culture. And this is not only true of HCT116s, it's also true of other cell lines where there's no contact inhibition and they can grow on top of each other. Okay, then we took a step up towards biological relevance by working with colleagues in Rolf Brecken's lab who do 3D organoid culture. So they did the organoid culture with the HCT116 cells. And here the effect is even more striking. everything's in the nucleus now. So this is exactly the opposite of what the people thought who'd been rejecting my papers and my grant applications by saying that Argonaute was always in the cytoplasm. In fact, here's a case where it's entirely in the nucleus. Okay. So let's take another step up in biological relevance. We obtained tumor samples from our tissue core, and this just is what happens in benign tissue, and it's mostly in the cytoplasm. In a tissue that was actually, the pathologist told us, was pre-malignant colon tubular adenoma, mainly in the cytoplasm. Okay, but then in 5 out of the 21 tissues we analyzed, we saw a shift to not only the nuclear fraction, but even when we isolated the chromatin, Argonaute was associated with the chromatin. So, you know, we now have a system that not in all colon cancer tumors, but in a fair number of them, RNAi factors like Argonaute have been sucked out of the cytoplasm and moved, relocated into the nucleus. So then the question becomes, you know, how is this changing what's going on? Okay, so let's just remind you of what we're dealing with here. The basics of what you have to understand is that this is a ribonucleoprotein complex that is programmable. You have an RNA domain, which is a microRNA. And you have Argonaute, which is a protein domain. Those bind together to form the ribonucleoprotein. So they could bind to a 3' UTR in a cytoplasm and do the canonical silence gene expression that you find in textbooks. But could it also be possible that they bind near splice sites to affect splicing? And could an effect on splicing be one of these effects that we're seeing in these tumor cells? And this would not be unprecedented. Anyone who's followed the antisense oligonucleotide world knows that the most successful antisense oligonucleotide drugs, the ones that are actually saving people's lives right now, helping kids to walk instead of dying when they're 3, are drugs that affect splicing by binding near splice sites. So we tried making some of those same drugs as double-stranded RNAs, and this just shows one result that was just in a simple model system showing how these double-stranded RNAs can affect splicing and do it just as well as an antisense oligonucleotide. So proof of principle, we know that a synthetic double-stranded RNA delivered by Argonaute can efficiently affect splicing. So the question we face is this very robust result, can it also occur with natural microRNAs? Okay, so what do we need as experimentalists, okay? This is something that is very difficult. And we've learned that even cytoplasmic RNAi, the thing that people thought was all settled and well understood, it's very, very difficult to find an actual, be able to say that this microRNA controls this gene, or this gene is controlled by microRNAs at all. Now we're gonna have to try to find out what's going on with splicing, something that's really new. So we need an efficient workflow to analyze the effect of increasing cell density on gene expression. We need to know how that increase in cell density affects splicing, and it's essential that we prioritize genes accurately before time-consuming experimental bench science, because I want my graduate students to graduate in no more than 5 years. I do not want them spending 8 years in the lab trying to chase down results. We need to be highly efficient. Okay, and that's where engaging with Almaden Genomics has come in. So this is very, very recent. I was in Boston for a conference only a couple months ago on antisense therapeutics, and that's when a representative of Almaden Genomics, which is also located in Dallas, met with me to talk about their platform and suggest that maybe we wanted to work together since we were in the same city. So everything that I'm going to be telling you about has advanced in the last 2 months, and actually primarily only in the last 3 weeks. Okay, so what does my lab need if we cannot work hand in glove with a computational biologist all the time? If we're going to do this and understand what's going on ourselves, there can't be coding. We just don't We don't code. Things should be drag and drop. Everything should be transparent, intuitive, and above anything, a pipeline needs to enable independent use by us. Okay, so if you have questions, I'm going to let Alban explain this a little bit more, but what I understand from this is that this is essentially like a Boston subway map. You may not understand every stop on it, but this provides a transparent map for the investigator about how the workflow is put together. And while I may not be able to understand it very well, because I can barely push the on button on my computer, this is something my students can understand. And this allows them to know what is going into this black box that ends up processing their data. Okay, but there's more. Is what they really like is the fact that through just a drag and drop mechanism, they can drag their different data sets over here, and then they can choose different outputs for how they want the data to actually appear. And I mean, and this is just marvelous compared to the way we've been doing it before. It gives them a great deal more power to fully utilize the data sets that we've spent so much time and money acquiring. Okay, so basically here's the experimental setup. We grow cells to different cell densities, 50, 250, 370, either day 3, day 5, day 7. We prep the RNA, we get it sequenced, and then we use their genome pipeline to analyze it. Okay, now remember, all of this has been done in the last just 2 to 3 weeks. There are various kinds of RNA splicing events. So the first thing they did was to look at various RNA splicing events and just put the different splicing events that are observed into the various piles. Okay, next we decided to try to narrow down which of those are going to be significant. And it's reasonable to expect that something is only going to be biologically significant if it occurs at both day 5 and day 7. That has to be consistent. It may miss a few things, but at least it cuts down the database a little bit. So there you go. We go from a couple thousand events to about 750 by doing that. Okay, that's the first step. Okay, the second step is we have data from a technique called eClip. Won't get into it in detail, but basically eClip is a sequencing technique that allows you to find out where Argonaute binds to RNA. The places where Argonaute binds to RNA are great candidates for where microRNAs are going to be binding. So this is really a very productive way of narrowing down your candidate targets. More than that, we only also demand that those have sites for highly expressed microRNAs. So that's 2 levels of stringency that we're imposing there. Okay, so now when we demand that those 2 things are happening, we've gone down to about 250 different events. So that's 250 candidates so far. Okay, then what do we do after that? Well, they've also, our Almond and colleagues have also been able to look at where are those Argonaute binding sites relative to an intron-exon junction, relative to a place where the splicing apparatus might bind and where you might expect that there be a disruption. And what we observe is that most of these are occurring within a couple hundred bases of these intron-exon junctions. So again, these are probably better candidates. Now, what we haven't had time to do now that Almond's pipeline has brought us down to a couple hundred candidates is what we would do is we would individually curate each one, look at the data, and just by eye and figure out, is this something reasonable? And this is something we routinely do. My lab always looks at the individual pieces of data, basically at the raw data stage. But their pipeline also makes that easy. Okay, so what else do you need to know about it? Comprehensive logging. There are full records logs for every part of the analysis, software versions and parameters. That ensures that everything is well documented, the results are traceable, allows researchers to repeat their analysis, and offers a checkable record of the process. There's containerization. Every study is marked with a version, keeps track of the software used. That's always a nice thing. They give a stable, repeatable setup for the software and analysis. And importantly, all the images in study are free for everyone. So when we go to publish our results, we're going to be able to put everything out there, because obviously we can't get it published if we don't give people the opportunity to reproduce it. Okay, finally, I'd just like to share what Crystal said. So Mark Kumatomi and I were having dinner last night when working on the final version of this talk. I opened up my computer and there was an email from Crystal because I'd asked her what her experience had been using this. And I showed it to Mark without actually knowing what Crystal was going to say. So Crystal could have said, this is garbage. This is a complete waste of time. I guess that would have saved me from having to come here and give the talk. But in fact, this is what she said. And she didn't write this for this. This is straight from what she's, a very talented graduate said. It is very straightforward how to upload and incorporate the data. The splicing pipeline module that they built is transparent and easy to modify. The user interface is clear and colorful. Lots of nice drop-down menus to zoom in, modify, and export useful data. So that's the experience of an outstanding graduate student with this pipeline. My view is that this is a direction that we as experimental scientists need to be moving in, and both to increase our productivity, to make sure that we're not turning out garbage, but also to make sure that we get the most out of these data sets that we have spent so much time collecting. And that in the case of, for example, data from patient tissue, is extraordinarily precious and difficult to reproduce, and that we really need to squeeze every drop of utility out of. Finally, I'd just like to thank the people in my laboratory, in particular, Crystal Johnson, who's been working with the scientists at Almaden to get something in time for this conference. And I hope you understand that it's remarkable how quickly this has come together for a novel protocol. And in my experience, when protocols come together quickly, that's usually a good sign that they're going to be robust and useful. Finally, thank you all for taking your lunch hour to listen to me. I would be happy to answer any questions. Thank you.
Hi.
Yeah.
Diego, have you looked at the response during confluence when these cells are growing up of line elements at all?
No, we haven't done anything like that, but what I can tell you is that right now we are working out methods and having some success of being able to isolate chromatin. And those should be going in for sequencing very soon. And needless to say, I'm very excited to know what's going to happen with chromatin-associated RNAs. So that, if I was going to project out in the next couple of years, suffice to say, I'm really excited about moving into that area and adapting some of these techniques for chromatin-associated RNAs. Yeah.
Anybody have any more questions? If you want to go up to the microphone in the middle there, that would be great.
That's a great presentation. I have tons of questions, but we can talk about that later. Just a quick one. So when you were doing the experiment with a cell line that is completely confluent, do you guys were looking at? Do you guys ever measure this in primary cells that they normally become when they completely get covered the plate, not like this transformed cell line, but a normal cell line, they normally get quiescent.
We haven't done that. I know Joanna Vidigal at NIH has been doing experiments like that and has been observing a shift into the nucleus. And she thinks that's involved in transposon silencing. And I know she has a paper in review about that. I think experiments of that sort are extremely exciting. You know, we have a small lab, so we're focused on what we can focus on. But I think that putting these cells into biologically significant states and then observing whether Argonaute is shifted in those states is an important thing for people who are versed in those systems to do.
Yeah, I completely agree. And the other thing I was thinking, when you show your 3D spheres, and you clearly see the, you know, the shifting, have you guys ever tried to put, for example, these cells in vivo, and then try to see if you see even the same phenotype?
No. I mean, we haven't tried to do that, though. I mean, it's certainly something that we can do now that we're currently writing this up, and that will close the first chapter. I think that's an obvious thing to do, not for us to do, but for our collaborators who did the organoid culture to do, because they do that all the time. Okay. It would be simple. Yeah.
Okay.
Great. No, thank you. Are there any other questions? Okay. Well, if not, I'm available. Oh, Leonard. We couldn't have one without you giving me a question. We have to make up for the years that we haven't seen each other.
Yeah, for the years of like not asking questions from the front row to each other. Yes, exactly. It's wonderful seeing you here, and thank you for, as always, for the stimulating, provocative talk. You know, this is great data. I've never seen any of that. I mean, the nuclear AGO2 in HEC 116 and in the tumor samples, especially the correlation with benign versus malignant. This is very impressive. I guess I'm kind of wondering two things. Do you think that nuclear localization of Argonaute, even though it's real and functional, is basically, it's a property of wacky over confluent or like stage 4 metastatic tumor cells that at that point have such diverged genomes and morphologies that they don't even look like a human cell anymore? Everything spills over into compartments that they shouldn't be in, and that's how they got nuclear Argonaute? That's basically one question. I have another
Let's ask
concern.
Do it one at a time, because that's an important question. Okay, my speculation is that when cells are growing normally, either in culture or in the body, RNAi doesn't do that much. I'm sure it does some important things like buffering expression and stuff, but not really anything outstanding that we're going to have an easy time finding. I think probably it's when you push them to more extremes, for example, in cancer, that things start to get regulated differently. So that would be my guess. For example, in development, you might think that there needs to be a little bit more regulation to make sure everything happens right. And that's when things might start changing. Also, I should say that one thing that we've also observed is when they move into the nucleus, you get de-repression of genes in the cytoplasm. So it's also possible that the effect is, there's nothing important going on in the nucleus. It's that you're reversing cytoplasmic regulation of gene expression. So those are my guesses right now, but I think other investigators have to look in other systems to really answer your question.
Yeah, exactly. So like, what are the larger implications of this? Is every component of the risk complex currently known to be nuclear, or aside from AGO2, it's still the jury's out on that?
That's a wonderful question. Okay, again, based on our data right now, TNRC6, the critical scaffolding protein that promotes cooperativity, its expression at high confluence seems to go way down. So that would suggest that it's not as able to be a scaffolding protein at high confluence. So what is the impact on that? That's something we're going to have to look at, but it appears that it can't form the high molecular weight complex as much. But then also, I don't want to get into the complexity of it, but there may be some differences also in the 3 TNRC6s and how they behave. So yeah, a lot of interesting stuff there.
Yeah, as we discussed earlier, there is a growing body of anecdotal reports and probably some literature about successful RNAi of allegedly nuclear RNAs, especially non-coding RNAs, which seems to indicate that unless they're somehow cytoplasmic and undetected, which is unlikely, especially when people look for them with fish, there seems to be a way for siRNAs to successfully reach allegedly nuclear transcripts somehow. And this is fueling speculation in the field about nuclear risk.
Right, right. No, absolutely. Although one thing that I will say is that I don't think there's any doubt that small RNAs can get loaded in the cytoplasm, imported in the nucleus, and then bind to targets. In our experience, the actual cleavage of those targets is much, much less predictable. So right now, imagine them working like binding factors, not cleavage factors like it happens in the cytoplasm.
Thank you. That's great. Very provocative.
Okay, thank you.
Thank you.
All right, are there any other questions? All right, thank you very much.
Thank you so much.